Okay, today we're talking about bacterial transformation and how it relates to the different units of AP biology. So to start off, the chemistry of life. The phospholipid bilayer that makes up the cell membrane is impermeable to the plasmid that's being added to the matrix initially. So in order to allow the plasmid to enter the cell needs to undergo a heat shock, which is going to split this cell membrane and allow it to be permeable to the plasmid. So if we imagine we have a little plasmid is going to go through and hopefully be able to stay inside of the cell. The reason this isn't possible beforehand is because of the chemical properties of the cell membrane. And as you can see, it's densely packed and cannot allow any large and polar molecules through, which plasmids are both of those. Now on to what is Unit 2 of AP Biology, the cell structure and function. Part of the cell structure is the plasma membrane, which we did talk about in terms of the chemistry of life and how those interactions take place. But the plasma membrane blocks the diffusion of the plasmid, as we previously stated. So the plasma membrane needs to be deformed in some way, which is the heat shock, to allow the plasmid to enter the cell. And then once it's in the cell, the plasmid is going to interact with different structures such as the ribosome, enzymes, all that. And so that's cell structure and function and how it relates to this. Now, in terms of cellular energetics, a big part of cellular energetics is enzymes. And as I kind of alluded to in the previous explanation, enzymes are a very critical component in basically all the processes which have to take place in regards to this lab. Enzymes are a necessity for the transcription and translation of the plasmid, and without those two things, this lab is pretty much useless in general. Enzymes also are critical for breaking down ampicillin. So once the enzymes allow the plasmid to be transcribed and translated, the translation actually results in the formation of new enzymes, and these enzymes are the enzymes that actually provide the antibiotic resistance and all that. Now, in terms of cell communication and cell cycle, cell communication does play a large role in this, but the cell cycle is, very obviously, it's what has to take place in order for us to have results that are visible. So the cell cycle, which is basically the replication of the internal components of the cell, including the DNA and including our plasmid, is necessary for the proper reproduction, or as you can call it, more of the binary fission of these bacterial cells, which allow us to create our little colonies of bacteria with this plasmid, and allow us to see a glowing plasmid under UV light, a glowing colony under UV light. In terms of heredity, all of the lineage of our initial cells, which were able to gain the plasmid, the cells in which the plasmid diffused into and stayed as the plasma membrane closed up on it after the heat shock. These cells were reproducing, and there were only these cells were surviving in the environment full of the antibiotic, because only these cells had the antibiotic resistance. And so as they began to replicate, only the cells with the plasmid survived, and thus eventually the whole entire lineage would have the plasmid, since that's all that could survive. And that kind of relates to something we're going to get to very shortly. But in terms of gene expression and regulation, the expression of GFP, as we were able to tell from our results, only takes place in the presence of lactose. So without lactose, the operon which allows for the coding of the GFP gene inside of the plasmid is activated by the presence of lactose. So lactose acts as a little activator, I guess. Yeah. And that allows for the GFP to be expressed and thus allows for our little colonies to glow. And then this is what I was alluding to a second ago. Natural selection, as we have a bacteria that are more suited to an environment, or they're more fit for that environment, they're going to reproduce at a much higher rate. And because of this high rate of reproduction, eventually the entire population is going to be filled with what is basically only the bacteria that contain these specific traits that made them more fit for their environment. So down here is a good little infographic. We start off with only a few resistant bacteria and a lot of non-resistant bacteria to the antibiotics. And eventually all these bacteria, which are not resistant to antibiotics, die. The ones that are continue to reproduce, and eventually we get from a population like this, which is all orange, to a population like this, which is nearly all red. And the red is the bacteria with the antibiotic resistance. Now, for our final unit of AP Biology, Unit 8, Ecology. To start, the growth of bacterial colonies is impacted by density-dependent factors. Density-dependent factors are a part of a microecology between bacteria. So this essentially means that, as bacterial colonies become more dense, they're going to reproduce, and they're going to reproduce at a slower rate, essentially. Now, the addition of the plasmid to the bacterial population creates a more diverse genome as well. Diversity is a big part of ecology. Diversity is what stabilizes populations in an environment. And through the addition of our plasmid, we essentially created a more diverse population. And so once the external factor of ampicillin was introduced into the environment, the cells which did not have the plasmid and were not resistant to antibiotics, or specifically ampicillin, they all died. And because we did have cells that were resistant, the population was able to survive and continue to grow. And so that is essentially the most important part of ecology. And that was our 8 units. So thank you for listening. Hope you enjoyed, and hope you like our little plasmid up here.